Hi, everybody. So thank you for joining us for today's presentation entitled Single-nucleus and Spatial Transcriptomics of Archival Pancreatic Ductal Adenocarcinoma after Neoadjuvant treatment. And it's my real pleasure to introduce you to William Hwang. He's a really fantastic young scientist based in Boston. He's the Holman Research Fellow at the Broad Institute and the Koch Institute for Integrated Cancer Research. He's a resident physician in the Harvard Radiation Oncology program at Mass General and Dana-Farber Cancer Institute, the Brigham and Women's Hospital, and the Boston Children's Hospital. And for a complete biography of William and all our speakers, you can visit the tab at the bottom of your screen. Now, if you have any questions during the presentation, please submit them in the Q&A box, and our speaker will respond back to you via email. So please join me in welcoming William. I'll turn the presentation over to him.
Hi, my name is William Hwang, and I'm a research fellow at the Broad Institute of Harvard and MIT and Mass General Hospital. I'm excited to be here today to share with you our work on single-nucleus and spatial transcriptomics of pancreatic cancer after neoadjuvant treatment. I have no financial disclosures. So pancreatic cancer remains one of the deadliest solid tumors. Despite several decades of concerted research efforts, clinical outcomes have not improved significantly. As you can see from the annually updated cancer statistics in the United States, on the top left, in men and women, the incidence of pancreatic cancer is only approximately 10th, but the estimated deaths attributed to pancreatic cancer currently ranks 4th and is projected to be the #2 cause of cancer death by 2030. And a large reason for this is that the other cancers ahead of pancreatic cancer have seen some significant improvements in therapeutic options and outcomes over the past several years. Whereas even for localized surgically resectable pancreatic cancer, the 5-year survival is only approximately 30%. And if you are diagnosed with metastatic disease, the 5-year survival is only a dismal 3%. So, as with a lot of other cancers, there is an interest in improving precision oncology for pancreatic cancer. And the first place people turned to was looking at what somatic mutations could tell us about a person's particular type of pancreatic cancer. Unfortunately, in one large study of long-term survivors with pancreatic cancer compared to those that had a more typical survival duration, exome sequencing didn't reveal any significant differences in the somatic mutation profiles among these very disparate patient groups. Furthermore, comparing primary and metastatic tumors shows a pretty limited mutational heterogeneity, suggesting that non-genetic mechanisms may actually be the predominant source of clinical variation. So as with some other cancers, researchers in the pancreatic cancer field turned to looking at gene expression, and in particular, doing transcriptomics in bulk of pancreatic specimens, generally in the treatment-naive setting. And there were 3 major efforts over the last decade by Collison, Bailey, Moffitt, and colleagues, where they looked at primary pancreatic tumors and they did unsupervised clustering of their RNA profiles to try to understand what subtypes might distinguish the clinical variation seen in patients. And there were 2 widely considered tumor intrinsic profiles known as the basal-like or squamous profile and the classical progenitor profile that are considered to be tumor intrinsic. And then in addition, there are some other subtypes that have been identified, such as the exocrine or the immunogenic, that have subsequently been found to be likely driven by stromal contamination. Part of the problem and the reason we're seeing this with the bulk transcriptomic data is because despite the aggressiveness of PDAC, the malignant cells can be in the vast minority of actual cells that compose a tumor. In fact, in some pancreatic tumors, we see single-digit percentages of the neoplastic cellularity. Most of the rest of the tumor is composed of various stromal components. So these various bulk efforts actually try to address this by either enriching for epithelial content by selecting specimens with more than 40% epithelial content, or by performing microdissection. And both of these lead to biases in the resulting data. So this seems like the perfect setup to move into the single cell RNA sequencing realm that has really provided additional insights in numerous cancers to date. The problem with efforts in single cell RNA-seq for pancreatic cancer is largely twofold, however. The first is the fact that these tumors are very desmoplastic, and therefore dissociating tumors into a single cell suspension is very difficult. And the more you dissociate, the more you compromise viability. So that trade-off has become a big problem for pancreatic cancer. The second issue is that the pancreas's normal role is largely a digestive role. Proteases, RNases, other digestive enzymes are replete in pancreatic tissue. So when you're trying to maintain RNA integrity for an RNA-seq readout, that becomes also very problematic. So we attempted to address these challenges by developing and optimizing a single nucleus rather than single cell RNA-seq approach, which we call SNUC-seq, which is useful in several regards. First, you don't have as many problems with the digestive enzymes because the specimens are snap frozen, and when they thaw for processing, the whole process only takes about 10 minutes from beginning to end in terms of extracting the nuclei and separating the RNAs of interest from the digestive enzymes. The second benefit of this is that dissociation becomes much less of a problem because you don't need to maintain cell viability. And then the last, potentially largest advantage of SNUC-seq over single cell RNA-seq is that you don't need fresh specimens. So we're able to take archival frozen specimens that were stored for a different purpose many, many years ago and actually obtain very high quality single nucleus RNA-seq data from them at the present time. So this workflow summarizes to date the 27 specimens that we have processed. About 50% are treatment naive and 50% have undergone neoadjuvant chemoradiation. And these are all surgical specimens that we're working with. And we split the specimens for SNUC-seq and then a subset for bulk RNA-seq, multiplex ion beam imaging, as well as Nanostring digital spatial profiling. And what we've noticed empirically is that an RNA integrity number using the Agilent bioanalyzer platform of greater than 6 seems to be necessary in order to have sufficient RNA integrity for good downstream SNUC-seq data. So this slide summarizes the 15 patients in the treatment-naive cohort. You can see there's a broad range of ages, of genders, of stage grade, margin status. A few of them have the more rare adenosquamous histology, and then also a pretty broad range in terms of adjuvant treatment and clinical outcomes. For the 12 neoadjuvant treated patients, we have a similar range in terms of their clinical pathologic features. A couple of the patients had BRCA2 germline mutations, and all of these patients got neoadjuvant radiotherapy. The majority got FOLFIRINOX as well. And then 2 patients got nivolumab or losartan on trial. So we processed all of these 27 specimens through our pipeline, and it yielded, after filtering and quality control, about 170,000 nuclei across the 27 patients. And we performed community detection using a Leiden algorithm, and we were able to identify distinct cell populations using known cell type specific gene expression signatures. And you can see at each of these boxes is a UMAP for an individual patient, naive on the left, treated on the right. And the gray cell clusters represent the tumor cells, the malignant cells. And then the various colored clusters represent different types of non-malignant cells, including acinar, ductal, endocrine, endothelial, fibroblast, immune, Schwann, and smooth muscle. So all the major cell types we were able to identify with confidence from the transcriptomic profiles in our nuclei. On the bottom right, those stacked proportion bar plots are illustrating at an individual patient level the wide range of cell type distributions in the naive and treated settings. So to further increase our confidence in calling the malignant cells within our tumors, we performed inferred copy number variation analysis. So this is just one example patient, and on the top of this plot, basically across all the chromosomes, you're looking at the inferred copy number variations by a sliding window of 100 genes, the expression of 100 genes sliding along each chromosome arm, and trying to see if they're concordantly enriched or depleted and inferring copy number variations from that. So on the top, this is from the normal adjacent pancreatic tissue from one of our tumors. And you can see there are really no patterns suggesting CNV clonality. And then on the bottom, this is the actual tumor itself. So some don't have any CNVs and are likely non-malignant cell types. And then you can see some distinct clusters marked in gray on the left that correspond to our malignant cells, where you see a very distinct CNV pattern. And then as a gut check, we wanted to see, since we only had 27 patients, were the CNVs that we were inferring from our 27 patients, were those representative of pancreatic cancer patients overall? So for this, we turned to the TCGA cohort of about 180 some patients, where they used SNP and whole exome sequencing to determine arm level somatic copy number variations. So in the plot on the right, I'm showing the CNV proportion relative to the chromosome arm. In dark blue is our SNP-seq data, in light blue is the TCGA cohort, and in gray is a different cancer altogether, that's prostate cancer. So as you can see, there is a pretty close association between the proportions of CNVs that we saw in our data compared to TCGA, and very distinct from the prostate cancer data from TCGA. So the next thing we wanted to do was to really test this hypothesis that SNP-seq gives us better representation of the cell type proportions present in the actual tumor in situ compared to the single cell RNA-seq approaches that have been tried prior to this. So for this, to give us our kind of histologic ground truth, if you will, we use multiplex ion beam imaging. We used a panel of about 27 different targets, but what I'm showing you here is the main compartments of interest. So the images on the top, on the left in red, that's CD45, and on the right it's CD31. And then green in both cases is keratin, dark blue is vimentin, and then cyan is the nuclear compartment marked by double-stranded DNA antibody. And on the bottom 4 panels, we used a software package called Histocat to basically to segment and create masks for each of these compartments. And then from that, we were able to enumerate the number of cells across 3 different fields of view per specimen. And then when we averaged those together and compared it with the cell type enumeration by SNP-seq, you can see for the 8 patients that we did this side-by-side comparison for, some differences, but for the most part, pretty close correlation between MIBI and SNP-seq for the proportion of endothelial, immune, fibroblasts, and epithelial cells. And then on the bottom right, those are 2 summary stacked bar plots, naive on the left, treated on the right, for all the patients combined. And you can again see a pretty close correlation. So this confirmed our hypothesis that the processing involved with SNP-seq seems to be able to better capture the cell type proportions present in these tumors. So next, we combined all of our 170,000 or so nuclei in the treated and treatment-naive cohorts and made combined UMAPs out of them. So gray, again, is tumor, is the malignant part of the tumor, I should say. And you can see that the tumor cells are segregating by patient as expected, whereas the non-malignant cell types are grouping together across patients. And you can also make a few kind of initial qualitative observations here. There's significantly more malignant cells in the naive cohort compared to the treated, and there seems to be an enrichment for some of the non-neoplastic cell types, which is better shown in this box plot here. The acinar, the ductal, the endocrine, fibroblast compartments are all enriched in the post-treatment cohort, whereas the tumor is significantly depleted, as you would expect, with some treatment response. And the increase in the acinar, ductal, endocrine, et cetera, cell counts after treatment is thought to be both related to potentially direct stimulation and regeneration of normal pancreatic tissue with treatment, but also a known desmoplastic contracture of the tissue after chemoradiation that just simply increases the density of some of these normal cell types that are relatively resistant to chemotherapy and radiation. So this plot is just to illustrate that, without dwelling too much on the specific details, that we can zoom in to, the immune population, the endocrine population, and use further known cell type signatures to refine these larger groups into various immune cell types or endocrine cell types. And from a pure enumeration standpoint, we see some interesting changes with treatment. For example, on the plot on the left, you can see that there's a significant depletion of Tregs post-treatment. And then on the right, I'm breaking out just the dendritic cells in the treatment-naive setting on the left and the treated setting on the right. And you can see that there's a significant increase in the classical dendritic cell type 1 or CDC1s with treatment, and a significant depletion in CDC2s. And this is so much the case that we were not even able to reliably detect any CDC2s in the post-treatment setting. And this is interesting because there's some preliminary data from some clinical trials looking at neoadjuvant chemoradiation and the effects on the immune system that suggest this same shift towards a CDC1 dendritic cell phenotype, which is associated with cross-presentation to cytotoxic T cells and more of an anti-tumor effect compared to CDC2s, which are known for their role in immunosuppression, wound healing, et cetera. So to go beyond enumeration and look at in detail what changes are occurring with treatment, we did a differential expression analysis comparing post-treatment specimens with treatment-naive specimens. So in these 7 volcano plots here, the right side of the plot represents genes that are enriched in the post-treatment setting, and on the left, genes that are enriched in the treatment-naive setting. So this is still an ongoing analysis at this time, so I'll just call your attention to a few highlights that were very robust findings. So on the top left in gray, this is the neoplastic compartment, and we found that chemoradiation was associated with an enrichment in type 1 and type 2 interferon response genes, but also a depletion in developmental programs previously associated with the classical progenitor subtype in bulk, which the COMPASS trial recently found was more sensitive to cytotoxic therapy compared to the basal squamous subtype. So our data seems to be consistent with those findings. Amongst the immune cells, I'll call your attention to the CD8 T cells, which are shown in lavender on the middle row, the right volcano plot. So the treatment was associated with upregulation of several activation programs, such as IL-7R, TCF7, STAT4, and SLAMF6, which have been linked to memory activation, cell survival, as well as response to checkpoint inhibitors in other cancers, such as melanoma. And concordantly, there was a depletion in the exhaustion markers TIM3 and CD39, which again, in melanoma has been associated with a lack of response to checkpoint inhibitors. So this was interesting to us because while checkpoint inhibitors have largely failed in metastatic pancreatic cancer, except in the 1% of patients that have microsatellite instability, these results seem to suggest that perhaps there's a different situation when you combine combine immune checkpoint inhibitors with chemoradiation in a neoadjuvant setting. And certainly that's something that several trials are currently looking into, and will be interesting to see what those results are. And then finally, on the bottom right, in that yellow volcano plot, those are the fibroblasts. And after treatment, there seemed to be this enrichment for a hyperactivated myofibroblastic phenotype. So the next thing we wanted to do was, use this opportunity where we have this almost 170,000 nuclei from 27 patient samples. We could really use this opportunity to kind of go beyond the basal classical distinction that has been well established in the bulk setting, and look at what other common gene expression themes are shared across multiple patients and might have some bearing on the biological behavior of this very aggressive tumor. So without going into some of the specific details, because we actually recently did a reanalysis of this topic modeling, I want to make the point that we do see, when we do topic modeling across our patients, an endoderm progenitor, and an epithelial topic very consistently. And those are very similar to the classical and basal squamous subtypes identified in bulk. We also confirmed findings by other groups that the same tumor will have cells that express a predominantly classical profile, whereas others will express a predominantly basal profile, and those are heterogeneous within the same tumor. But beyond that, we see other topics that are also very prevalent, mesenchymal, neuroprogenitor, et cetera. And we're in the process of doing this topic modeling, not just for the tumor cells, but also the fibroblasts and other cell types, and really trying to learn what themes may correlate or predict outcomes in this disease, and then taking what we learn at the single nucleus, single cell level, and using that information to deconvolve bulk RNA-seq data that's much more readily available and translatable into future clinical trials. So this slide is just showing that if you classify a patient's individual cells based on the dominantly expressed tumor or fibroblast topic, you can see there's a broad range in terms of the heterogeneity and distribution of different topics within each patient tumor. So we were able to learn a decent amount, and we're still learning stuff with further analyses in the dissociated setting. But we're really limited by the fact that the tumor has lost all spatial information at this point. And a lot of the important interactions between different cell types, or even between the same cell types, can only be captured if you have spatial fidelity and resolution. So we partnered with Nanostring to use their digital spatial profiling platform known as GeoMx to help us get transcriptomic information that's spatially resolved and really take the data to another level. So what I'm showing here is a schematic of the Nanostring workflow for their GeoMx platform. And there's two very similar paths you can take. It's just different. The difference is just the probe. You can go after protein targets or RNA. We mainly focus on RNA using ISH probes, but you could very easily do the exact same things with antibodies. So these antibody or RNA ISH probes have been tagged with a barcode that has a UV cleavable linker that attaches them to the probe. And we start with an FFPE slide from the clinic, and we do all of our standard processing, dewaxing, antigen retrieval, et cetera. And then once everything has been processed, we stain that slide using a combination of 4 morphology markers to label the nuclear compartment and 3 other morphological compartments of interest, and then a probe set from Nanostring that covers your targets of interest. And then after you label that, you put the slide onto the device, and there's a slide scanner built in, and you scan it, and you see the 4-color immunofluorescence image, and you're able to then go in and choose specific regions of interest, or within those regions of interest, areas of interest that you want to interrogate further. Then the digital micromirror device that Nanostring developed will customize the illumination mask to fit whatever your region of interest or area of interest looks like. And it'll actually move to that area of the slide, illuminate, and photo cleave all those barcodes in that area of interest. And then those will get sucked up by the straw and deposited into a spatially indexed 96-well plate. And depending on what plex your panel is, you can either read out that spatially indexed transcriptomic information on the nCounter platform or with NGS. So with Nanostring, we first started with their Cancer Transcriptome Atlas, or CTA panel. This is a carefully curated over 1,500 RNA target panel that was collated from various other well-established cancer signatures that are listed here. And here are 3 of our samples that we have performed this protocol on. I'm showing side-by-side images of the 4-color immunofluorescence and the ROIs that we selected next to an H&E image. And what we've labeled in green, the epithelial compartment, in magenta, the immune compartment, and in cyan, the fibroblast compartment. So the question that we were really interested in answering that requires spatial resolution is, within the same tumor, we expected that there would be regions that were infiltrated by immune cells and regions that were exclusionary to immune cells. And that there may be tumor intrinsic or potentially fibroblast intrinsic transcriptional programs that were in part governing this local immune microenvironment. And we wanted to understand what that was with the hopes that that would lead to improvements in immunotherapy strategies in this disease. So what I'm showing here is the 3 different types of regions of interest that we see across most of our patients. On the left, this is an example of tumor cells that are surrounded by fibroblasts in cyan, as well as immune cells in magenta. The middle panel shows tumor cells that are only surrounded by fibroblasts. And then on the right, these are tumor cells only surrounded by immune cells and no fibroblasts. So the Nanostring GeoMx platform has really nice auto segmentation feature built in where you can control some of the parameters like thresholding and erosion factors. But ultimately, you get really nice segmentation, as you can see here, where we actually went after very complex patterns, based on what our tumors look like, to actually separate out the tumor, the immune, and the fibroblast compartments and interrogate them separately within each ROI. So we collected our data, and because this was over 800 plex, we had an NGS readout, and we took that data. The first thing we wanted to do was to see how it compared to the SNP-seq data. So we actually took the SNP-seq signatures for various cell types, and we used those to deconvolve the Nanostring data. And as you can see in this heat map here, we got a very clear epithelial cluster on the top. The green ROIs represent epithelial, and you can see it really matches the tumor signature from SNP-seq. Similarly for CAPS, this would be 1, 2, 3, 4, the fifth row down. You can see there's a very clear fibroblast signature that correlates between the Nanostring and the SNP-seq data. And then various immune populations also correlated very well. So this really demonstrated nicely the coherence and complementarity of the Nanostring platform and the single-nucleus RNA-seq approach. So next, we wanted to compare the differential expression patterns in the tumor and fibroblast compartments by treatment in the Nanostring data compared to the SNP-seq data. So here you're seeing a volcano plot where genes enriched in treated tumor AOIs are on the right, and genes enriched in naive tumor AOIs are on the left. And without going into the details, since this is just a preliminary analysis I'm showing you here, a lot of the initial genes that we're seeing are comparable between the two platforms for both the tumor and the fibroblast, which was very reassuring to see. So moving into the spatial realm further, we used our data to ask the question, what genes are differentially expressed in the tumor compartment based on whether or not the ROI is infiltrated by immune cells or not? And we were able to get pretty robust signatures from this that we're still currently exploring. But this is data that we could only get with spatial resolution and not with the dissociated data. So we're really excited to explore this further. And then we can also do something very similar with the CAF compartment to try to derive fibroblast intrinsic programs that may govern the immune infiltration in the local environment. So with topic modeling, I mentioned earlier that we were able to move beyond the traditional classical versus basal distinction and really look at a whole host of themes that various tumor cells within a pancreatic cancer use to potentially gain a fitness advantage and collaborate for that goal. And from a very initial look, we only have 1,500 or so genes in the CTA, which is a lot of genes, except when you're looking at the topics, those are derived from the whole transcriptome. So quite a bit of imputation has to occur because they're not all represented in the CTA panel. But even with that limitation, when you look, for example, across the 8 topics that we identified in the cancer cells, we found some interesting associations. For example, the mesenchymal topic was strongly associated with ROIs that contained fibroblasts. And then looking at it in a different way, we found that the endoderm progenitor, which is similar to the bulk classical subtype, was associated with immune infiltration more than other topics. So these are things that we're currently exploring more, but certainly yielding some interesting results already. So to address, this limitation, as well as really open the doors to a lot of other potentially interesting questions, Nanostring has recently developed a whole transcriptome assay that is really changing the way we're able to do this analysis, because we no longer have to impute certain genes in our topic signatures. We now have all the genes available. And we did a preliminary analysis with them that so far is showing some exciting early results. So this is the 4-color immunofluorescence from the Nanostring GeoMx platform. And you can see some example ROIs that are overlaid on the image. And we found that across the transcriptome, we were able to successfully detect 6,600 genes spatially with our samples. And this is without optimization, just a very initial pilot. And what I'm showing here is in these joy plots, the top 3,330 most highly expressed targets by segment. And the colors correspond to what we've been using. So cyan represents the fibroblasts, magenta is the immune, and green is the epithelial. And to see if we were on the right track, if you zoom in on one example fibroblast, AOI, you can see that the scores after deconvolving for our topics are much higher for the fibroblast topics compared to the tumor topics for this fibroblast AOI. And conversely, for the epithelial AOI, the tumor topics are much more highly expressed than the fibroblast topics. So this was consistent, with what we would expect, and suggests that the data is going to be very useful as we move forward. So we're very excited at this point, with the preliminary data that we have, that between the better understanding of pancreatic cancer molecular taxonomy, as well as more insights gained looking at the effects of neoadjuvant chemoradiation and other treatments, we're really going to be able to move forward with the spatially resolved technology and start to understand the interplay between the various components of these pancreatic tumors, and ultimately be able to inform precision oncology and pancreatic cancer in a much more meaningful way. So thank you very much for your attention. I hope you found the presentation useful. I'd like to thank several people without whom none of this work would have been possible. First and foremost, my postdoctoral advisors, Aviv Regev at the Broad and Tyler Jacks at the Koch Institute. Very close collaborators who have contributed to every part of what I've shown you today. Are Karthik Yagadish, a computational biologist in the Regev lab, Jimmy Guo, a UCSF MD PhD student, and Hannah Hoffman, a research technician working closely with me. I'd also like to express my gratitude to my collaborators at MGH, Brigham, and Dana-Farber, especially Andy Liss, Carlos Fernandez, and Mari Mino for the clinical side of this project. And then also my close collaborators at Nanostring, Dan Zollinger, Kip Furman, and Jason Reeves for all of their help. And then last but not least, the funding agencies that have made this work possible have been the Lustgarten Foundation and the Concla Cancer Foundation of ASCO. Thank you.